Assalamualaikum warahmatullahi wabarakatuh, and a very good day I bid to everyone who are watching this video. So this is Biogenesis 2021, a virtual final seminar of mine. My name is Nur Atika binti Zulkifli and my metric number is 192077. I was previously supervised by the late Dr. Bukhari bin Zainal and now is co-supervised by Dr. Muhammad Nur Hisham. My FYP title is Cellular Structure Changes of Pouteria campechiana fruit during growth and development. This is my presentation outline for the day. Okay, so introduction. Pouteria campechiana, or commonly called canistel or egg fruit, or in Malay we call it as buah telur, they possess these four characteristics, which is showy, attractive, flavorful, and nutritious. However, they also come with some problems. They are underutilized. So the four reasons why they are underutilized is that they are underestimated of their own potential. There has been inadequate research on their commercial exploitation. They lack knowledge on their food and nutritional potential. And lastly, it is unavailable to find their complete botanical information. However, Baehni and Charles 2006, they claim that canistel deserves wider planting in order to become a good ideal addition to the botanical information and also cellular structure inventory. I've come up with the objectives of this study, which firstly to observe Pouteria campechiana at different fruit development on cellular level, and number two is to analyze the carotenoids level in canistel fruit according to its fruit development. The significance of this study is to tackle Sustainable Development Goal put up by the United Nations, which is the second and the third goal, which is zero hunger and also maintaining good health and well-being. So as a justification, this research was conducted in order to increase the knowledge on fruit development of this fruit and also to become a reference to find any changes of carotenoids level, so then we can come up with a good inference on how to tackle quality of fruits. Moving on to literature review. So for this part, I believe that there are three elements that are important in my study. The first one is canistel fruit, the second one is scanning electron microscopy, and the third one is carotenoids, which is in the next slide. For the first part, canistel fruit, this is actually a table of maturity index put out by Sunila and Murugan 2017, where they list out these parameters that can be considered for us to classify the fruit into stages. From my field of study, I chose to do stage 1, stage 3 and stage 6 because they provide a very different morphological changes in between them, so then clear inference can be made. Next, this is scanning electron microscopy, which is a device that I used to find the micrography of the tissues for Pouteria campechiana by using the principle of focus beam of electrons. Lastly, which is carotenoids. In fruits, we know that the green and the yellow coloration is actually imparted due to the lipid-soluble chlorophylls and also carotenoids present in the plastids. Other than that, carotenoids is important because it is associated with the lowering of latex properties, increasing sugar content, losing acidity and astringency, and lastly, a decrease in texture due to flesh softening. Moving on to the materials and methods for this study. So Pouteria campechiana can actually be found in Bukit Expo, Taman Pertanian University in UPM. The region of this study is actually consist of 4 canistel trees there. To assess the fruit of Pouteria campechiana, I had randomly tagged around a total of 200 inflorescence. The harvesting was done when they reached stage 1, stage 3 and stage 6. Next, for the first objective, which is to undergo histology study by using scanning electron microscopy, this is actually a series of procedures in order to do the SEM viewing. This whole processes is used to decrease the amount of moisture inside the fruit and also to increase the polarity of the fruit. The second objectives, which is to analyze the carotenoids level inside of this egg fruit, this is the steps to do spectrophotometry in order to find the means of carotenoids level on 3 different developmental stages, which is the stage 1, stage 3 and also stage 6. Next, results and discussion. From this figure, it is found that the harvested fruits of Pouteria campechiana from stage 1, stage 3 and stage 6 have provided different morphological features in between them. So first one, which is size, the circumference and the length has increased gradually due to the active cell expansion. Next, for the texture, from my personal experience, it is hard at first, but then it softens up when it reaches stage 6. This is due to the pectin presence where it undergoes flash softening and also loss of structure. Lastly, for the color, it is very conspicuous that the color changes from green to bright yellow due to the chlorophyll breakdown and also carotenoid synthesis. This is a post-cutting and handling on the mesocarp. For the first stage, we can find that there are small dots of latex on the mesocarp, while on stage 3, the latissifer increases and it becomes the most abundant amongst the 3 stages as the fruit when oozing with latex liquid. It went sticking itself on the board and the blade. And then for stage 6, the latex presence was far lesser than the stage 3. Okay, for the ultrastructure of Pouteria campechiana on stage 1, which is 4 weeks after anthesis, as you can see in A, the cell has an initial beehive formation, or to be specific, it is a pattern called angular polyhedral, which means roughly spherical. The cell was then covered with latissifer in the form of dust-like particles surrounding the primary cell wall, labeled as L. Next, stage 3, which is 12 weeks after anthesis. On A, we can see that there is an excessive embedding of the latissifer to the point where we cannot see anymore the polyhedral pattern. Latex presence in the fruit results into an astringent texture in the mouth. This would be an unfavorable experience for the consumer, while B and C ultrastructure shows that there are some large lipid body. Lipid body can be indicated by the spherical shapes and numerous indented cells that are formed because of they are bumping with some organelles, and it will affect their smooth contour. Lastly, for stage 6, 24 weeks after anthesis, we can find that from the figure A, the mesocarp of collapsing cell wall inside the yellow circle revealed that there are some wrinkle and folded deformation. This happened because of the fruit firmness loss inside its pectin. The most obvious changes that we can see here is that they happen to loosen and break down of its cell wall and also cytoplasm swelling. Next, a decrease in the latissifer because we can see from the ultrastructure there are less dust-like particles that are found. This is also an indicator to the maturity of the fruit. Then, for the intercellular spaces, we can find that it is very prominent on stage 6 due to the loss of moisture and also transpiration phase of the stomata cells. For the second objective, which is carotenoids of Pouteria campechiana, we can say that from the figure, there are some prominent differences that can be seen in terms of color. Then it is also supported by table 2, where we can see that there are some significant difference in between stage 1, stage 3 and stage 6 in terms of their carotenoids pigmentation. It is found that the least carotenoids pigmentation level is at stage 1, while the highest one is at stage 6. Thomson had once claimed that the fruit color was one of the primary external aspects that will influence the consumer purchasing decision. Therefore, it is very conspicuous that from these results, stage 6 fruit has the most attractive color, and it may become a catalyst to please the canistel fruit consumers' appeal. A simple analogy was brought by Wolf, where he said that animals are attracted to attractive colored fruit so much that they have become the biggest seed dispersion vectors. This is because animals can only divide their vision into dark versus light or blue versus yellow. So it is also the same with human perception on buying an attractive colored fruit. As for the conclusion, I believe that there has been some discovery on the canistel fruit cell pattern, cell wall disassembly, and also the latex composition. For the carotenoids, it is found that the carotenoid level increases significantly as the fruit grows. Lastly, for the recommendations, I believe it will be good if we can do cellular structural study by using other ultrastructural study apparatus such as TEM and also light microscope. I think that is all from me, and this is my reference. And with that, I say thank you.